Proteins give you more than just a kick in six-pack. They are especially vital to the structure and function of cells in the human body. Humans produce them at the ribosomes, or rather in the cytoplasm to be precise, during a process called protein biosynthesis. Two steps are necessary when it comes to the production of proteins. First, transcription, followed by translation. The information or blueprints necessary to produce the proteins are stored in the cell nucleus in DNA form. So, how is this information communicated from the nucleus to the ribosomes, where the proteins are produced? A closer look at the transcription process will give us a clearer idea. The blueprints are stored in the DNA. But how does all that work? After all, doesn't the DNA only contain base pairs? Well, that's exactly what the blueprint is. The base sequence, that is, the order of these base pairs, contains the genetic information, or rather the protein blueprints. The original blueprint is contained in the nucleus, the DNA, and makes an intermediate copy from it. This copy is then transferred to the ribosomes, which uses this copy to produce proteins. By the way, why not take a look at our extra video to refresh your knowledge of DNA? The intermediate copy is used to transfer the genetic information from the nucleus to the cytoplasm. Ribonucleic acid, or RNA for short, is precisely this transitional form. Real sticklers for accuracy will point out that it's actually called mRNA. The M stands for messenger, which makes sense as the mRNA is the messenger that brings the genetic information to the cytoplasm. Like a pizza boy delivering pizza from the pizzeria to you at home. Just to recap the important information, mRNA is used to transfer the genetic information from the nucleus to the cytoplasm. As we explained earlier, the production of mRNA is called transcription. The enzyme RNA polymerase carries out this transcription. You can pretty much compare it to a worker who checks the blueprint and creates a temporary copy. How does this enzyme work? Well, let's take a look now. DNA contains a whole host of blueprints for many different proteins. These sections of DNA that form the blueprints are called genes. So, for example, this here would now be a gene, which contains the blueprint for a certain protein. This gene is then translated into mRNA during the transcription process. A gene starts with a promoter. This short base sequence tells the RNA polymerase, here's where we get going with transcription. Then, at the end of a gene is a terminator, who tells the RNA polymerase that it's the end of the line. So, the RNA polymerase looks for the promoter and starts to translate that section of DNA into mRNA. It first unwinds both DNA strands to make sure it doesn't get all tangled up at the end, a bit like your headphones in the pocket of your jeans. It subsequently dissolves the connections between the bases to separate both strands. The nucleus is filled to the brim with free nucleotides, and these now begin to connect to the correct bases. The RNA polymerase now travels along the DNA. Wherever it separates the DNA strands, free nucleotides will connect to the bases. This continues until the RNA polymerase reaches the terminator. There, it separates once more from the DNA and the mRNA is finished. Okay, not entirely. This was just the pre-mRNA, a precursor to the mRNA. They need to be modified a bit more by eukaryotes, but more on that later. Altogether, the three steps were initiation, elongation, and termination. During initiation, the RNA polymerase attaches itself to the DNA at the point where the promoter is located in order to subsequently unwind the DNA and separate both strands. During elongation, the enzyme travels along the DNA and the nucleotides connect to the free bases. The elongation process ends with termination. The RNA polymerase removes itself from the DNA and the pre-mRNA is finished. And this making of the intermediate copy is called transcription. A few more important things to know. The DNA strand that is used as the template for the mRNA is called a coding strand. It encodes the mRNA. It's also called the antisense strand. The opposite strand is called the sense strand. You guys should also know that the transcription process starts at the 3' end of the strand and ends at the 5' end. Whoa, wait, the what now? This just relates to the structure of DNA. To put it in layman's terms, one end of the strand has a chemical group attached to the 3rd carbon atom, an OH group to be precise. The 5' end then has a group hanging from the 5th carbon atom. This is the phosphate group. This 3' and 5' end business is related to the structure of DNA. It helps us determine the direction of the transcription process, nothing more. As you can see, both DNA strands run in opposite directions. The 5' end of the one strand opposes the 3' end of the other, and vice versa. This is called anti-parallel alignment. When it comes to nucleotides, there's also some pretty important stuff you should know. The base pairs are always adenine, thymine, and guanine, cytosine. But things are a little different with mRNA. Here, thymine is replaced by uracil. From now on, every time you see uracil, you'll know that you're dealing with mRNA. Generally, the mRNA strand looks almost the same as the old strand of DNA, with the only exception being that it has ribose as its sugar instead of deoxyribose, and that the thymine base is replaced by uracil. Now, the pre-mRNA needs to be turned into the correct strand of mRNA. We'll explain this process in the next video on RNA processing. So let's recap. During the first step, initiation, the RNA polymerase shows up to unwind the DNA, after which the strands of DNA are separated from each other into 2 single strands. Then, the enzyme travels down the coding strand during a process called elongation. And now, the following happens. The free nucleotides bind to the bases, which are now free, to form the new mRNA. Well, as you already know, it's actually pre-mRNA. When the RNA polymerase reaches the terminator, we call this termination. There, the elongation process ends, and the RNA polymerase detaches itself from the DNA. And your pre-mRNA is ready. If you feel the urge to just keep on studying, then take a look at the video on RNA processing.